The Theoretical and Computational Biophysics seminar. It's good to see everyone in the room again. It's my pleasure actually to introduce Dr. Tony Auerbach from Department of Physiology and Biophysics at University of Buffalo, SUNY Buffalo, upstate New York. Very brief introduction. Tony was trained as a chemist initially at Cornell, and then in biology at UC Santa Barbara before he did some neuroscience work, and then he joined the University of Buffalo as a faculty where he went through the ranks. And as you will see, Tony has done tremendous work actually using electrophysiology techniques in ion channels, understanding the kinetics, the chemistry, what happens to conformational changes of the protein. He was one of the pioneers actually thinking around those, right? Because everybody thought things are instantaneous, you open the channel and that's it, and so on and so forth. So he is now here to tell us a little bit more about some theoretical and computational works that they have done in this domain. And you are very welcome actually to join the project that is going to present. If there are any students who's interested here to join this project, please come talk to me, and Tony is going to be here for the whole week to talk later. Okay, thanks for coming all the way, and looking forward to your seminar.
Now, one thing from my bio, I started my career at the University of Illinois.
Oh,
I was an NSF summer science scholar.
I
For
didn't
some
know.
reason, they posted me to Champaign-Urbana, and I was in the Burnside Research Labs.
Wasn't in your CV.
It wasn't in my CV.
No.
But I started with lipids, and that's what got me interested in membranes and ion channels. So the title is Agonist efficiency, zipping up the AChR, and just for today, let me see if I know how to do this. We're going to change the name of this group. We're going to put another letter in there for he, because this is going to be experimental. So I'm going to be talking about free energies, but the free energies are real ones. They're not computers. These are ones that come from experiment. So just for today, we'll add an E, although there'll be a little bit of T and C. This is the outline of my talk. There are going to be 4 sections. The first section is going to be about receptors, and that will be non-controversial background information, and I'll introduce the idea of efficiency there, because that's a new idea. The second part is going to be about efficiency in acetylcholine receptors. And the third part will be about a reaction coordinate that I think is very important, that I call catch and hold. And the fourth part is going to be about the zipper. The zipper is the mechanism of conformational change that I think the way the acetylcholine receptor changes conformation. So the receptor part will be non-controversial. It will be general. The stuff I will say applies to all receptors, if not all allosteric proteins. It's background information. The part 2 and part 3, the data comes from acetylcholine receptors, but there is some data suggesting that it is generalizable. Part 4, the zipper is from a very specialized data set, and I can't say if it's general or not, but given the track record of the acetylcholine receptor being a typical protein, I'm going to allow the possibility that it could be generalizable. So after each section, I'm going to give a little section for questions, although it's going to be a long seminar. So if you don't have a pressing question, just wait till the end, because I want to get to the zipper. Otherwise, I'll have to cut that at the end. Okay, so this is a nicotinic acetylcholine receptor. It's a ligand-gated ion channel. It's an intrinsic membrane protein. It's about 300 kilodaltons. There are many subtypes. It's very important in the nervous system and in the immune systems. Pharma is very interested in developing subtype-specific ligands for these receptors without much success. The type that I'm going to be talking about is the type that sits here at the nerve muscle synapse. So this is the nerve and this is the muscle, and here are these little dendrites coming up from the muscle, and they're stained in black is the acetylcholine receptors, and it's stained with a cobra toxin. So you know the acetylcholine receptor is important, because if it stops working, like with the cobra toxin, you stop breathing and you die. So I'm talking about the neuromuscular type, and that type, all nicotinic receptors have 5 subunits arranged around the central ion channel. The neuromuscular type has 2 neurotransmitter binding sites in the extracellular domain that are essentially identical and operate independently. During development, there's a subunit swap. Early in development, there's a different subunit, and in that case, the two subunits are different, but they are still independent. They operate independently. And down in the transmembrane domain, the pore narrows into a region that's called the gate region, and this gate region can either be dry or wet, depending on the global conformation of the protein. That's taking place about in the middle of the transmembrane domain here. So this is the most important slide of the seminar. You can leave after this slide. This is the core of the seminar. Receptors are binary proteins. That means they have two native states. Each state has a different output. It can be off or on. And each state has a different sensitivity to energy coming into the system. It can be either weak or strong sensitivity. So that's shown here in this potential energy surface. The energy landscape, the two native states are shown as energy wells. We're going to label them in the tradition of ion channels, C and O. C means the gate is dry, ions can't get through. And O, the state is wet, which means ions can get through. So this is closed and open ion channel. That's based on the outputs. And each of these two global states, these are global conformational states of the protein, can have C binds agonist weakly and O binds agonist strongly. So that's the setup for receptors in general. In the acetylcholine receptor, nature has selected a set of side chains that are much more stable on the whole in C compared to O. So it's an uphill reaction going from C to O. The probability of being open without any agonists is about 10 to the minus 6. And this is just each side chain gets selected almost independently, and all the energies add up. And most of the side chains are more stable at C compared to O, so the channel is usually off. Situation changes when agonists are bound. You can see that the agonist provides some stabilizing energy to C, that's the low affinity, the weak binding, but more stabilizing energy to O. And when you have two agonists bound, I have it drawn here as one, but for two agonists bound with two binding sites, the uphill reaction becomes downhill because of that extra favorable binding energy from the ligand, from the agonist. So it's so much so that with two bound acetylcholine molecules, the probability of being open in the open conformation is now 0.96. So it goes almost from 0 to almost with 1. So you can think of an agonist as another side chain that's unusual in two respects. Number one, it's not covalently bound to the protein, so it can come and go. It's about the same size as a side chain. And number two, it's one of the few side chains that is more stable than O compared to C. And it's a lot more stable than O compared to C. See, this is it's much more stable. It loves O. And I can only think of one side chain that's almost as good as an agonist. You can make a mutation. You can make one mutation that almost does this. Wow. But there's only one. Most mutations have a smaller effect. Okay. So this is the rule. Agonists turn on receptors because they bind more strongly to the ion chain. I think that's generally true. That should not be controversial. That's why agonists do their job. There's another state in this diagram, which is this barrier in between the two, and lurking somewhere inside this barrier is a point called the transition state. And at the transition state, the agonist has the same binding properties as it does in O. You can see it's giving you a lot more stability. So that means this big mountain you have to climb gets much smaller when there's agonists around. So the channel opens now much faster than it not only does it open with a higher probability, it opens much faster. It's about 10 million, you get about a 10 million fold increase in the opening rate when you put on two acetylcholine molecules, and it's only about a 4-fold change in the closing rate. So if you have a receptor and it opens quickly when you put on your agonist, it's changing this barrier region to the transition state 2. But we're not going to talk about that. That's part 4. In parts 1, 2, and 3, we're going to talk about just these two blue bars. The energy of binding to the closed state, energy binding to the open state. And these two blue bars determine agonist properties. The agonist properties are affinity, how strongly it binds, efficacy, how much it activates once it's bound, and this new idea that I'm going to talk about is efficacy. All come from those two blue bars. So we can take those two states and 4 arrows, those 4 states and 4 arrows and put them in a thermodynamic cycle and give equilibrium constants to each of the processes. The vertical lines are called gating, and horizontal lines are called binding. There are 2 affinities, but the word affinity is usually applied to the weaker affinity because this is the main activation pathway. So just quickly, there are 2 binding sites, so you would have to extend the model by doing that if there were 2. And not only that, agonists can do receptors can do other things. They can be desensitized, and there's 4 different kinds of desensitized states, and they can be channel blocked, and they can go into weird states that you don't understand. So there's all sorts of other things in cells that can take place. We're not interested in the cellular response here. We're interested in the mechanism of the molecular mechanisms of that one protein. So what we do is we can remove those from our signal. We do single channel recording, so they have a distinctive lifetime, and we can get rid of those guys. And we can, not only that, we can knock out one binding site either by making a mutation or adding a toxin. Well, and so if we get rid of desensitization, we could decide this is the main activation pathway. We could analyze that pathway very easily. Or we can knock out one site and just look at this, get a protein that just does this. And we can analyze all the rate constants and therefore all the equilibrium constants in there independently. Okay. Now, this is a thermodynamic cycle, and in a thermodynamic cycle, getting across the diagonal is independent of the pathway. This is the conservation of energy. It doesn't matter where you go this way or this way, as long as there's no external energy coming into the system. And that leads to this green equation here. The ratio of these 2 constants should be the ratio of these 2 constants. And people have argued this. There is energy. There's an ion flux coming in. The membrane is bending. You're in a cell. Maybe things are being metabolized, doing things. So they said, you can't use this bottle. Well, we went and we measured all these things completely independently. And for 4 different agonists at 3 different kinds of binding sites, it worked. Okay. This green equation is true. So there is no significant external energy coming in. Okay. You can ignore it. Everything's in thermal equilibrium. So everything happens spontaneously, which means driven by temperature alone. All you have to worry about is KT. And this is the simplest system you can think of. 2 equal binding sites that operate independently because we know they're independent because if we measure the energy at one site, measure the energy at the other site, you add them up, you get the same as so they're not interacting at all. So this is a very pure, clean system established experimentally. So it's possible to measure all those rate constants and using single channel recordings, but most receptors are not amenable to that. Single channel recording is a sort of special technique that's only useful with certain channels, and it's not generally applicable. So we wanted to generate what I wanted to do is generate something that's generally applicable, and that would be a dose response curve. So everybody knows what a dose response curve is. And as you increase the concentration of your agonist, the response increases until it reaches an asymptote. And these curves are characterized by only 3 parameters. You have the high concentration asymptote, the low concentration asymptote, and the midpoint. So there's a maximum, a minimum, and a midpoint. The word efficacy is the difference between the max and the minimum. But since the minimum is the same for all agonists, usually just the maximum determines what the efficacy word is. And potency is another word for EC50. So this is sort of energy coming in and energy coming out in some weird transform. So it turns out from those 3 easily measured parameters, you could just stick a ruler on this thing and measure. So this min is hard to measure, but max, you just stick a ruler, you can say it's 0.7. And here's this is whatever this term, we fit it to a Hill equation, but it's just an arbitrary thing. You just want the min max, the maximum and the EC50. And if you know the minimum gives you the gating equilibrium constant in the absence of agonists. You can use these simple equations, you can calculate your equilibrium constants and use the green equation to calculate the high affinity binding constant binding to O. But that's hard to measure. The minimum is hard to measure because it's almost 0, and it's very hard to measure something that's almost 0. Just like it's hard to measure something that's almost 1. So you want something that's in between. So I'm going to show you later on that you don't need to measure the minimum. We worked out a way that just from the max and the midpoint, you can calculate these 2 things, and these 2 things, these 2 constants are all you need to know to measure efficiency. Which when I'm going to be talking about efficiency. So here's some real dose response curves. with 3 different agonists. This is the neurotransmitter over here, and it sits in these vesicles in the presynaptic neuron. Here are those junctional folds in the muscles stained with the black stuff again with the cobra toxin. It's a blow up. This is a frog toxin called epibatidine, and this is tetramethylammonium. And these are the dose response curves. So we measure these dose response curves, and we do know the minimum. We took about 10 years to measure that minimum. So that's what this number is here. This one, in this case, we know the minimum, so we're going to put it to use. So that's the opening, that's that P open in the absence of agonists that I told you about, 10 to the minus 6. And from these things, so we get the midpoints, we get the maximum, and we can calculate these 2 binding equilibrium dissociation constants. And it goes from the micromolar range to the nanomolar range for the high affinity. And if you take the ratio of those 2 in that green equation, that's called the coupling constant. And these are the coupling constants for these 3 agonists. So this is the affinity is here, and this is the efficacy is really the coupling constant. So we can get all this stuff from a dose response. Now I'm going to talk about efficiency. This is new. You're not going to see this in a pharmacology textbook. Everything I told you before is standard pharmacology. Efficiency of a machine is energy out over energy in. And receptors are molecular machines that convert chemical energy from binding into mechanical energy for the operation of this valve that goes from wet to dry. So the efficiency of a receptor, of an agonist at a receptor, is the fraction of binding energy that is converted to gating energy. So this is a chemical energy to mechanical energy conversion process. So what is the efficiency of a receptor? Well, it's very easy. It's surprising no one did this before. The energy out, let's start with the energy out. That's just this. This is the energy out. This is the gating with an agonist versus without. That's the coupling constant. You know, energy is minus the log of the equilibrium constant proportional to that. So the energy out is this numerator. It's the log. It's the difference between the high affinity and the low affinity binding constant. That's the log of this ratio here. And the energy in is just how much energy is coming in from the agonist. Well, that's the high affinity binding constant minus the log of that. If you don't like that, I have another way of deriving this later on. But if this equation, it would be the energy, would be the equation for efficiency. That's eta. That's the standard thermodynamic term for energy efficiency. So we end up with this pink equation that the energy efficiency, the efficiency of the agonist is 1 minus the log of the low affinity binding dissociation constant to the high affinity binding dissociation constant. And this turns out to be the energy efficiency of a thermodynamic cycle, of a steam engine. This would be T hot over T cold, would be the energy of a Carnot cycle engine. So this is the same form of the equation. So this is the, so we go back to these 2 blue lines. Affinity is just the length of those lines. There's 2 affinities. There's the weak affinity and the strong affinity. Efficacy is the difference between those lines. That's been known for a long time. Efficiency is the ratio of those 2 lines. So you have the length of the lines, the difference of the lines, and the ratio between the lines are the 3 parameters that every agonist, that every receptor. This is totally generalizable. So let's go back to our dose response curves. And here they are again. Here's that table. And now we're going to look with an eye towards efficiency. And you can see this is sort of energy in. And you can see that ACH gives you more energy out compared to epibatidine. So ACH is more efficient. You just look at it, you can say ACH is more efficient. And similarly here, these 2 guys have similar outputs, but this guy requires less energy coming in. This is KD. So you would say that TMA is more efficient than epibatidine. But let's run the numbers, and this is what we get for efficiency. For those 3, yes, ACH is higher efficiency than epibatidine, and TMA is higher efficiency than both of these 2 over here. And so for every agonist at every receptor, this is totally universal. You have 3 things you can get from each agonist. You get the affinity, you get the coupling constant, which is the efficacy, and you get an efficiency. So these 3 things are completely generalizable. Efficiency is a new one. You'll never see efficiency written in a paper anywhere. So this is a new one. And so I just want to quickly go back and we did this through these single channel measurements where we selected out all those, got rid of desensitization and everything. What happens if you do this in a cell? So this one, we just expressed our receptors in a cell and measured the whole cell response. And that's the upper set of dose response curves. You can see they're not exactly the same. And we normalized all responses to this response here of AChR. So every cell we did a test pulse and then measured what the response to the different agonists were. And we get efficiencies that are not identical, but the relative efficiency, they're about 3% less here. So you can do this in a whole cell. You don't need to do single channel measurements. There's going to be a little more error in a whole cell, but it's not going to be very big. But this is the one that's more accurate. So we're going to use the more accurate one. So this is a first pause. I have two pauses here if anybody has a question. Okay, but remember, every question you ask is going to make me go longer. There's going to be a price to pay.
So, what is the difference between efficiency as a meter and potency? Potency, I know that's relying on the concentration of the ligand. Which one? If there are two ligands, as far as I understand, if there are two ligands, the lower concentration of one of them having the same response is going to be more potent. So what is the difference between efficiency and potency?
Potency is, as far as I know, potency is another word for the midpoint of a dose response curve. So you're more potent if your dose response curve is left shifted. If you have two agonists, if you go back here, ACH is more potent than epibatidine because it's left shifted. So it has a higher potency.
That means can we say like the more potent is more efficient?
No.
Or?
Because this TMA is less potent than epibatidine, but higher efficiency.
So how this meter will benefit the pharmacologist, like in developing the drug? Like they will
Sit tight. I'm just getting started.
Okay.
I got a ways to go. Now I'm just giving you the background on those two blue bars is what I'm talking about. If you believe the two blue bars, then everything I said is a no-brainer. Okay.
My second question is, so is this term would be applied only for the drug that is
So the
not,
lights are off
for the side that is
on
not
his microphone.
interactive
I don't see any light on his microphone.
O and C are independently active.
Each site has its own
Should I turn it off?
efficiency, affinity, and efficacy.
I'm gonna put it.
And you have two sites and they're not interacting, you just double the energy. But if they interact, you just add the two energies. So, and if they interact, they're going to get some coupling constant between the two. So this, we're right now, I'm trying to get one molecule, one agonist on one binding site, keeping it clean as can be. You can get more complicated for sure. And you had a question?
Yeah, I was just curious, does the efficiency correlate to the toxicity of the compounds that you're studying?
Does it what?
Does the efficiency correlate to the toxicity of these compounds?
Toxicity?
So as in, you showed the frog toxin, for transformation.
Yeah, it correlates with structure. It's much more interesting. No, the frog toxin, no, it's not related to that. Okay, it doesn't have to do with the fact. Okay, let's move on. First experimental result that needs an explanation. We have three experimental results that are begging for an explanation. 2012, we made this explanation. We observed this. That if you make a plot, if you measure KDC and then you calculate this in the thermodynamic cycle, given the other two equilibrium constants, you see that there's a linear relationship between these two binding constants, the weak binding constant and the strong binding constant, energy. This is log of the constant. These two binding energies are correlated linearly. And it doesn't matter whether you have a very weak agonist. This one only has an efficacy of 5%. This one, ACH, as I said, is 96%. This one is a very low potency, 4 millimolar. It is way to the right in the dose response. This is 40 micromolar, way to the left. So over this gigantic range of affinity and efficacy, each point here is a different agonist. There's something similar about all these agonists. These agonists belong to the same tribe. Okay. Now this was a surprise. when we measured this, and no one had ever measured this before. And the reason it was a surprise is because what we had expected was, this is what allosteric theory says, that the strong binding energy must be more than the weak binding energy, but not on a straight line. We'd expected points to be scattered all over the place here. All allosteric theory says is that you get more binding energy from the active state compared to the resting state. And that's where the motivation for the ligand activating channel comes, that this blue line is longer than this blue line, but it didn't say that they should have the same ratio. And we didn't know at the time what this meant. So in this paper, we waved our hands around a lot, but what we now know what it means is this slope is the efficiency. All of these agonists have the same efficiency. So agonists can be classified according to efficiency. They all have the same efficiency, regardless of how strongly they bind or how efficacious they are. And not only that, for all these agonists, the slope of this line is about 0.5, which means that the agonists double. The agonist binding energy doubles in the open state, is double in the open state compared to in the closed state. So here's another animation. The lines get bigger and smaller. The difference between the lines get bigger and smaller, but the ratio between those two lines stays the same. This was an unexpected finding. So that led us to another analytical approach. It turns out if you take the green equation and the pink equation and combine them, you get this equation here, which is an equation for a straight line, it's blue equation, where the slope of the line is a function of the efficiency. M is the number of, M is the slope of the line, 2 is the number of binding sites, and this is the efficiency. Y equal mx plus b. So if you make a plot of log L2 on the y-axis and log 1 by kd on the kdc on the x-axis, you should get a straight line whose slope gives you that. So I'll show you all those agonists now. So here's that plot. Here's the same data. Well, it's a different set of data, but because it's done for each binding site separately, but replotted in that form. So log L2, which is really energy out, not the response, that's the energy out, right? It gets the energy out of the system. That's how much you're getting out. And this is the energy you're pouring in. This is an energy plot. Is a straight line according to that efficiency plot. And all these points fall on that straight line to give you an efficiency of 51%. So all these agonists have 51%. And these agonists are like ACH, this is nic, this is DMPP, these are choline derivatives, anabasine, carbachol, nornicotine. All these agonists have the same efficiency, even though they have different scaffolds. Nicotine and ACH are different. This is a quaternary mean. This is not, okay? They're different compounds, but they all have the same efficiency. So that's interesting. But what's more interesting, so efficiency is interesting that these all have, but what's more interesting, what happens when we plot some of these other guys? You remember epibatidine here was 42%. Well, we measured epiboxidine, anatoxin A, azobicycloheptine, cytosine. These guys all have a bridge nitrogen here. These two guys do not. This is tetraethylammonium, this is tetramethylphosphonium, okay? But these guys all fall on a 42% line, okay? And this TMA was 54%, I remember that, 54% for that little guy. Well, we took these two guys here, DMP and DMT also were 54%. Now, these were all fitted independently, these three lines. These are straight lines that were fitted independently. They all went to the same point, which is L0, which is the same L0 that we got by other methods, that's 7 times 10 to the minus 7 that we took 10 years to figure out. This is an easy way to get that number. You just take 3 agonists that have 3 or 4 agonists with the same efficiency, and you plot them on a straight line, and you go to the 0 intercept, and you can figure out your L0 that way. So this, as you may or not remember, this came from, these two things came from just the maximum and the midpoint of that dose response curve. You did not need to know the minimum. So each one of these points is a dose response curve, a full dose response curve. So there's about 35 dose response curves in this plot. There's a lot of data in this plot. So the N classes are discrete and precise. There is a real difference between 54 and 51. I'll show you a slide that proves that later on. So there are agonists belong to different efficiency classes. Now everyone here should be thinking like, well, what's the basis of that? Why are these guys 42 and those guys 51? What makes an agonist belong to a certain efficiency class? It's an obvious question. But before we get there, I'm going to show you one more. There was one agonist that we measured. This is varenicline, it's a smoking cessation drug. that did not fit on any of these three lines. It was over here, but we knew L0 now. We used the same number, and we calculated the efficiency at 35% for that line. What I will say here is that this correlation between, L2 was efficacy. This was L2, and this was KD. So this is affinity, and that's efficacy. The correlation between those two, which is clearly apparent in the data, is only apparent when you have a lot of points. If I were to do these three agonists here, I would come to the conclusion there is no correlation between affinity and efficacy. In fact, that's what everyone would say. Every pharmacologist would tell you there is no correlation. And that's because of this kind of data. And similarly, here's agonists that have the same efficacy but very different affinity. So if you only looked at a few of these, you would not see it. You have to look at a lot of data. Every point is a full dose response curve. You have to understand how much data is in this thing. So if you're doing dose response curves with agonists, don't stop at 3 agonists. Don't stop at 6 agonists. Do 20. Okay. Okay.
So the blue one we just connect.
The blue one L0 is the same for all agonists. That's the gating without any agonists.
I see.
So that number is the same for all agonists. So I just drew that. We're now searching for the blue ones, but I'll show you that blue is another class. So there are discrete efficiency classes. So we went to the literature and we pulled up some of the dose response curves where we could get the efficacy and affinity. And this was a good one from Trevor Smart's lab in the University College. And we used statistical methods to identify outliers. These are statistical methods. We didn't pull this out. And we fit what was left, the blue lines, to give you these efficiencies. So this is, and these give you the zeros, which are about 10 to minus 6, 10 to minus 6. So they're in a reasonable range. So, but what I want to point out this muscarinic paper in particular, because they say in this paper that here's 2 agonists that have very different efficacies but the same affinity. So they say there is no correlation between affinity and efficacy. I would tend to say there's another family there. These 2 guys happen to have big rings. They have structurally different than these guys here. Same thing with here. These 2 guys have sulfur. These are the only 2 agonists that have sulfur atom in there. So I don't know about these other ones. So I think, and the other point is muscarinic receptors, you're not looking at a channel opening. You're looking at something downstream. You're looking at G protein activation of potassium channels, and you would say, boy, that's not going to have anything to do with efficacy. Well, I think it works. I think it works very fine with GPCRs. You don't have to have an ion channel. You can do this with any receptor, GPCRs, and I'll give you the reason for that later on. Okay, now this is my interlude to go into the computational part. I don't know, how much time do I have? Really, I used up 48 minutes already?
No, you have 25 more minutes.
Okay, good. Then I'll go quickly over this because this is not my main activity, but I'm put it in there because this is a computational group. So this is the neurotransmitter binding site of nicotinic receptor. It says K of 5 aromatic residues. I'll talk about this glycine up here that's conserved, and this is called loop C, this loop over here. So, and I won't go through the numbers on these things here. So first thing we did was we said, well, what's the size of these? There seems to be an inverse correlation between efficiency and the size of the head group. Okay, so this is the ranitidine, which we took the head group here, and this is the head group here. This is antitoxin A. For the head group here, we took this and we took one more methylene here. And this is the size of the head group for whatever we got until we got to a rotor. We took 1 rotor plus and we measured it, and we just used Chimera. We didn't do MD simulations to figure out the volume that's being swept. So it's not a very accurate measurement of the head group volume. But we took that, and there looks to be some correlation. So big agonists have lower efficiencies. It looks that way to us. To go along with this, we measured the size of the binding pocket, those aromatic binding pocket, in both the open and the closed conformation. I'm going to go into this in detail if anyone's interested later on how we did this. And And we did this either by geometrically measuring the size of the pyramid formed by the centroids of those 5 aromatics, or we did it with a program, I forget what it's called. What's a PVLOE? There's some.
There are so many of them.
Yeah,
I know what
it
you mean.
uses water probe to figure out what
Right.
a binding pocket is. That's
Right.
what this one is. But in all cases, in general, we always found that the open pocket was smaller than the closed pocket. Okay, and that the extent, so that, and while the closed pocket was the same for all agonists, the extent of the contraction or shrinkage was greater with higher efficiency agonists. So the efficiency was correlated, contraction was less with low efficiency ligands, is what we found. And so our hypothesis, which I no longer is correct, but I'll put it out there, is that the van der Waals contact limit the contraction. So the contraction is somehow coupled. It's important for the launching energy into the conformation. For the chemical mechanical transduction involves a contraction of the pocket. And that contraction is less when you have a big agonist in there because of van der Waals bumping. I don't know if that's true or not. That's a testable hypothesis. So we also calculated the MMP-BSA energies for 2 agonists, for ACH and epibadidine. And we got these 2 efficiencies, which are not accurate, but they're in the right, at least they're in the right order. They're both about 25% lower than the experimental value. So I just wanted to show that to you. This is using, this is the PV structure that we used for that. So, okay, so that's my computational part. Okay, and we'll see whether that's useful or not later on. But now I want to get back to those binding site, because we made mutations of those residues and asked what are the effect of mutations of those aromatic residues on efficiency. So these are all 5 aromatics that were mutated. There were 44 different mutations in this plot. And we see that some of the mutations didn't have much effect, and all of those, or one of these, didn't have any effect. But there was another population that showed up, which is 55%, which you will recall that was 55% was the wild type binding site for those small guys. So particularly of the tryptophan 149, they were all in the 55% population. And then there was one over here that was 35%. So that's what we got when we mutated them. And actually, these experiments were done long before we knew about efficiency. We recalculated efficiency. Then there's that glycine up there. We did 4 different agonists at 4 different mutations of these guys. And these was all with 51 or 54% wild type agonists. They all dropped down to 42%, which you will recall is the same as epibadidine. Okay, that was what that red was, 42. These guys started 55, 51, they went to 42. And there was one that went to 32, which is similar to that blue one. And we've done this with two other mutations not shown there further away. And we drop it down either to 42 or to 35 or 32, something like that is what we get when we make mutations of the binding site. So that says that the values are not only discrete and precise, they're also modal. Our hypothesis is that the distribution of efficiencies is like a spectrum. It's like a line spectrum for a binding site, or a barcode is what I'm using here. That each binding site is characterized by a discrete spectrum of efficiencies. And that's because our hypothesis is that when they go from C to O, they can only adopt discrete structures. It's not a rubber room. The binding site doesn't bend and it goes from click to clack or click to cluck. It's not all over the place. That the binding site has discrete output structures, O structures that it has, and each structure has a different efficiency. It can go to a 55% or a 51% or a 42% or a 30% efficiency. We think that's an important observation. When we look at that gamma binding site, we get different efficiencies. We're starting to get a barcode for that. It's that for that other binding site that we have, the blue one that I showed you in the beginning, instead of the 2 red ones. The blue one, I think, is going to have a slightly different spectrum barcode. And then one of these is a disease-causing mutation that reduces the neurotransmitter efficiency to 42%. That's in here somewhere. So it's a possible mechanism for disease. You would think a machine that has a lower efficiency might be disease.
Mhm.
And it's possible that diseases are caused by a change in efficiency, just like modulators, which are molecules that bind away from the neurotransmitter binding site to affect function. Those guys could do so by affecting efficiency. So it's another way to think about diseases and modulators. Okay. So let's get back to something that's general. That's generalizable to all of them. So here's our blue equation again. And if you know efficiency and log is and the unliganded gating, then you can calculate one of these from the other, which means you can calculate an EC50 from a maximum and a maximum from an EC50. So what's you say, well, what's the good of that? You have to know those two. Well, L0 applies to all agonists of the receptor. You measure it once and you're done. We measured that 7 times 10 to the 7th 10 years ago. I use it every day. It's the most useful thing you can have is that number. You measure it once and you're done. So eventually you're gonna have L0 for your receptor. And N applies to many agonists. It applies to a class, and maybe it can be predicted from structure, not only from the MMTPSA calculation, but also for if you have nicotine and you put an R group here, since the head group is determining efficiency, you're probably gonna have the same efficiency here, but you can have widely different efficacies and affinities. So you get a lot, if you know efficiency, you know it applies to many different agonists. But what this means is that if you just measure just one concentration, the response at one concentration, max, you just take these circle ones, and you know the efficiency in L0, you can calculate the entire dose response curve in just one measurement. You don't have to go scanning anything. You just go to the high concentration, measure the response. I say, I know L0, I know the efficiency. You can calculate the entire dose response curve. That's what these lines are calculated. And these are real dose response curves. And similarly, if you know the EC50, a lot of people normalize their responses because they don't know how many channels are in the patch or in the cell.
Mhm.
So if you just take the potency, the midpoint of the curve, and you know your efficiency and L0, you can recalculate the entire dose response curve. And you can do this from the literature. You can take there's millions of these things published in the literature. And if you know the efficiency, you can recalculate, find the efficacy of all these compounds, which you did not know the efficacy for. And this shows you it's very sensitive to the efficiency value. So this is if we use for TMA, if we use 55%, we get the right result. If we use 50% for TMA, we get a very wrong result. So that's further proof that those two numbers, that 54% was really real. So affinity from one concentration and efficacy can got from a normalized dose response curve. Okay, that's the end of part 2. Okay,
So,
wait. She was
okay, I'm sorry, go ahead.
Is there a correlation between the head group size and the binding affinities?
No.
Because there's one
Only efficiency, not affinity.
has to be the ratio.
It's the ratio of those two lines. Yes.
So when you do the MMPPSA, you
Yeah.
know the open structure and closed structure
Yeah.
for a particular ligand.
Yeah. We
Right.
dock the ligand into an open and closed structure. We do.
Right, but
docking.
that's you have a closed structure or ligand bound structure with acetylcholine, for example. You don't have it with the other ligands.
No,
So you
no.
don't know the
We
contraction.
don't have it with either. You have it's way worse than that.
Oh,
Okay.
okay.
I'll talk. We can talk to you about it later, but we have to make some assumptions in calculating the CNOs. There's a closed structure
Okay.
with a toxin involved. We have to take out the toxin
I
and dock
see,
it.
I see. Okay.
Okay, and during the MD simulations, the structure switches, and it switches from closed to
Gotcha.
open. So we get two structures that way.
I see.
So there's a lot going
Fair
on.
enough. Okay, you talk.
So you mentioned that modes, discrete barcode, correspond to distinct post conformational states.
I don't know if it's a hypothesis.
Okay. Do you think that means that we'll have this barcode predictive of currents?
I don't think it predicts what's going on at the gate, if that's what you're asking.
Yes.
I think it's a very local thing. I think the PAR code is predicting a discrete structure of the binding site that is adopted with different class agonists. So everything in the red class goes to one site, one form. Everything in the blue class or the green class is going to go to a red binding, or that color open structure is the hypothesis. I would love someone here to test this. This is a computationally testable thing, but I have no idea. These are just based on my experimental observations. Okay, we're going to go into the third one, which is short, and then the last one, which is the zipper. So here's the second unexpected result. If you measure binding of ligands to the binding pattern, and you can do it to the C state and the O state, as I showed you way in the beginning with that thermodynamic cycle, what you find is binding to C is slower than to O and very agonist dependent, whereas binding, this is the on rate, the association rate counts, and binding to O is close to the diffusion limit. Okay, so now, why is this a surprise? Because people see in structures, they see loop C come over the binding site when you have a strong agonist in there, and they say, oh, loop C covers the binding site. It's a lid. Loop C traps the agonist in the binding site. I won't show this very well.
Don't show the agonist.
No. So they say, so this is what everybody expected, that the binding to C should be by diffusion. You read in papers. Agonists diffuse into their binding site. You see it all the time. It's wrong. They don't diffuse. C is slower than diffusion. And we expected O to be slower than diffusion, but this is what you see. This is the real data. It's for 4 different agonists at 3 different binding sites. This is the pattern. And where is Claudio here? Claudio's not here.
It's on Zoom.
Claudio's on Zoom. Okay, well, Claudio.
Here I am.
Okay,
Yeah.
there he is. Claudio made something like this observation in a very elegant way, way back in 2001. But now we have the real numbers for it measured directly. And so the question is, what's going on? So in order to go on, what we're doing is we're pulling a page from Michaela Smith and saying, let's look inside these arrows for binding and gating. So if you look inside the arrow this is the binding step. A plus C goes to AC. That's the binding of the ligand to the closed state. To break it down, there's 2 things going on in that. There's a diffusional step that agonists have to diffuse to the binding site. And then there's a conformational change, is what the hypothesis is, that is different for different agonists. And that conformational change not only gives the agonist dependence, it also slows it down. So if you notice back in this one, that the rate, this is potency. This goes back to your potency question. This is the same region. This is the trend of potency, okay, or KD actually. So the more potent, the faster it gets in. And that's because this conformational change happens more rapidly with stronger agonists, is the hypothesis. I think that's a reasonable one. So diffusion is the same for all the agonists that we're talking about have approximately the same diffusion constant. So the big agonists, it'll be different. The question then becomes, well, why is open at the diffusion limit? So we know it can get in there at the diffusion limit. It's not like you're waiting for the agonist to fold up in a certain way. The agonist can get into the open pocket. That's the smaller one. The smaller pocket with loop C covering it is the faster it gets in there. Okay. So loop C is not a steric barrier. That's I think everyone knows. And in the gating reaction, we're going to break that down in detail in the next section. But there is this shrinkage of the binding site, which is the efficacy. That's the coupling constant there. That's the switch from low to high affinity. And then there's the rest of the protein isomerization. And both diffusion and the rest of the isomerization are not influenced by the agonist. The only thing the agonist cares about is what's happening at the binding site. Things are local. And so and I'm going to give these names. This conformational change I'm going to call catch. And that's a local rearrangement of the binding site that lets the agonist in and results in weak binding. And if you want to see pictures of my fantasy of what that could be, that I have a slide afterwards to show you what that. It's a cartoon, no data. And the conformational change switches the I'm calling hold, where the pocket contracts and gives you small binding. So catch and hold is the reaction coordinate that counts when it comes to the agonist. Don't talk about binding and gating. We're talking about catch and hold. So when I make this efficiency plot of KDC, that's the delta G of catch. And I make this of log L2, which is the gating. That's L2 is the whole thing. As far as the agonist is concerned, that's a delta G of hold. So each of these axes has an offset, which is the diffusional offset and the isomerization of the rest of the protein offset on this axis. But it's the same, offsets are the same for all agonists. So this is, you can translate this whole plot up and down, and left and right. Okay, but you're going to get the same slope, the same efficiency. And I think this is why GPCRs work, muscarinic receptors work, because there is an offset that's the same for all agonists after the catch. There's catch and hold, and everything else that happens afterward is the same for all agonists, and therefore you can get the slope right, you get the efficiency right for those things. Is that clear? I'm getting a puzzled look on people's faces from this. Well, I'm going to plow on because I'm running out of time. So everyone should be saying, "Ha, this is interesting. Delta G catch is linearly related to delta G hold. An energy change in binding is linearly related to an energy change in gating." Now, everybody thinks of binding and gating as separate processes, and here I'm telling you, they're not separate. They're related energetically. There's an energy link between binding and gating. You can't say, "Oh, the thing binds and the thing gates." They're the same thing. Binding and gating are the same thing. This is very weird. This is a very weird thing. Okay. So here's how do you explain this? Well, here's binding and gating. Here's the catch part. Here's the hold part, which are the only two things that vary with the agonist. They're related by a linear free energy relationship. So this is the reaction coordinate I'm talking about. It's catch and hold. It's one reaction coordinate, so that you have energy change in catch and an energy change in hold. This is the sum of the two. Again, this ratio is the efficiency, gives you the efficiency when you have a strong agonist. When you have a weak agonist, you just don't tilt it as much. So you have the same efficiency. This ratio stays the same for choline, for a weak agonist. Okay, but you get this catch and hold linear relationship. So this energy diagram is what accounts for that the plot, the efficiency plot that I showed before. Is that clear? This is getting complicated now. Okay, we're getting some head knowledge.
Yes.
That's okay. It's just free energy. It's just this, you have to link the two energies. Binding and gating have to be linked energetically. I'm linking catch and hold by this tilt of a linear free energy relationship. So, and what different efficiency means is that this point is just moved left or right. So it's moved here. So you can see here, this is the fraction of energy under catch, and this is the total. So if you move this to the left, you get more under the hold part, which means higher efficiency. So that's the other way to derive this equation. If you do, I won't go through it. I think you believe that equation. So I won't go through it. So so binding and gating are really the same thing. And I'm calling binding gating, I'm calling it a sweep. I didn't know what to call it. So, and this is where the zipper comes in, beginning of the zipper. So this is how I think happens. So you have a ligand shows up. I have little cartoons at the bottom. It lowers the transition state for catch. The ligand, by structural matching, lowers the transition state for catch as the hypothesis. Like an enzyme. I'm basing on enzymology here. And then you get the catch. You lower the transition state and the end state, and you get the catch conformational change. And once catch happens, catch lowers the transition state for hold, and then hold happens. This is the structural basis. I see someone shaking their head here. That's the structural way you would get a linear free energy relationship between binding and gating, is if you had this series of changes. And so binding and gating crosstalk, and here's the zipper. So that's how a zipper works. So what a zipper works when you form this link, it takes this transition state and lowers it to that. So you get the next one. So the agonist shows up, it lowers the transition state, which was here, it was here. That transition state was high. These were far apart. And then because that agonist showed up, it lowers it down into this state over here. And that allows the next link to form, which is down here again. And then that lowers the next transition state up here, right? And then that causes the next link to form, and so on. So that's why I call it a zipper. A zipper is a visual representation of this reaction coordinate linkage, this linear free energy relationship between binding and gating is called is a zipper. Now, I don't know if you may not like the zipper word, but that's why I use the word zipper. And of course, this is detail balanced. If catch promotes hold, you can see here by that mechanism, after you form this pre-catch complex, you don't hold. The second step doesn't happen. You have to do the first step in order to get the second step in the zipper. You don't form the link in the absence of the zipper of the previous step. The previous step promotes the formation of the next step. That's catch promotes hold. But you can see by detailed balance, I guess I'll skip this. I'll skip this. But it's true. Hold promotes catch. It has to happen. This is thermodynamics. So catch promotes hold, and hold promotes catch. I'll use that in a later slide.
Yeah.
Talk about the wave in the next slide. I'm going to pause there before I talk about the full zipper. So let's take a breath. That's a little hard. I see some confusion.
I was thinking, if you can imagine some conformational, some structural relationship for the catch and hold.
Oh,
That,
yeah,
you know, catch and
Yeah.
hold and
yeah. That's a great question.
it's understandable, but do we know anything about the structure that?
We know hold is a contraction of the pocket. Okay, and we think loop C coverage, movement of loop C, we know certain things about catch,
Right.
about hold. We can compare the two structures in the closed and the open with the ligand bound. We don't have the APO structure, and when we do MD simulations of APO, it's moving all over the place. So we don't have a very good picture of that. But you would think that if they're energetically linked, and they should be structurally linked.
Yeah.
But it's not necessarily the case. Energy and structure are not always so simple. It's possible you have a structural event that is energetically linked to a different structural event in some way that I don't know. It's possible. Although I agree with you, that's my first thought would be whatever happens in catch probably happens in holds to a smaller extent. Yeah.
That promotes it, whether it's, you know, break of hydrogen bond, formation of hydrogen bond, and water movement,
Something. So yeah, the transition state, knowing the transition state between those things, that would be a fabulous. One of the beautiful things about this receptor is that things are local. You can take a 20 angstrom radius domain and calculate stuff and not worry about what's going on way down there on the membrane and everything else. So I think this is a doable computational event, is to understand those two, catch and hold. That would be a good question. Okay, now we're going to do the zipper, the full zipper. Everybody's zipped up. And not zipped up. He's ready to go. He wants
Preparing
to
my
go.
question at the
Okay.
end.
This is the third unexpected result. So I'm showing you results that have to be explained. These are solid experimental results. And what this is the energy of the transition state relative to the energy of the end state. So I'll show you how we get these, okay? And this is the organization of the transition state for gating. So we're measuring now that at the transition state, we're asking how much does it resemble the open state? Remember, I showed you that the agonist, those two lines were the same for the open state and the agonist. So the agonist at the transition state was essentially open. So now we're going to say what it's like all over the protein, not just at the binding site. So I'll show you how we get these plots later. So again, before we were looking at these two blue bars. Now we're going to look, whoops, I turned it off. Now we're going to look at these two purple bars, which is not the well, but the barrier versus the open state. We're going to measure the energy of those two sites. That's what this next plot is about. So the way we get that is we make a bunch of perturbations. So here's different agonists. Here's a whole bunch of agonists, including water on this plot. Okay, and we make a plot of the opening rate constant. Like this is the transition state, the barrier height energy, and this is the well energy of the final state, the end state. Okay, this is, and you can see that, and this is for the agonist, if we change the agonist, and this is if we change this agonist. This is, we change this to every other side chain, okay, 20 different side chains here. And this is what we measure single channel, we measure the rate constant, and we measure the equilibrium constant for all these different constructs. And we get the same plot, same slope, 0.93. So the transition state is almost the same energy change as the open state. Not quite, it's 93% as much. Okay, for that's at this site right here. That's for just right here, these two sites. Now we do this over and over again. For these are different, each one of these plots is a different set of mutations of one residue. This is one residue, all these different mutations, we measure the open and closing rate. So we do it again, and we do it again, and we do it again. We do it again. We do it for, this is 3,000 mutations, all agonists. This is why I mean it's an unusual data set. Nobody did this. Nobody was crazy enough to do this. I was crazy enough to do this. And this is the histogram that we get. So this is 3,000 mutations, probably 100 and some residues. I don't know how many. So each one of these bars, one bar is one plot of what I showed you before. We have hundreds of these plots. Each plot has 5 different runs. So there's, like I say, thousands of these things there. So what is it, that value? That slope is called phi or phi. And so there's 5 distributions. And what does that mean? Well, this is what it means. This is the theory part. Phi gives sequence. So this is a sequence of, in this case, it was 6 transitions if you perturb the second transition, either by making the equilibrium constant smaller or bigger, that's what these dashed lines are. You measure the rate constant. This is from simulation. You measure the rate constant and then the equilibrium constant, you plot them versus each other. You get this line here with the black dots. And if you did the third one, you get this line, and the fourth one, you get this line, etc. So this gives you the phi value you would get as a function of which reaction you perturbed in sequence. So phi gives you sequence. That simulation was done here. We've also done the mathematics. You can solve this mathematically too. This doesn't have to be done by simulation, even though people told me I couldn't do it because it's a 5th order polynomial. You can do it. Okay. So phi gives you sequence. And so we think that in the transition from C to O, there are 5 intermediate, short-lived intermediate states. And we associate each one of these colors with the phi value that we measure from this distribution. And we give them a name. I'm calling hold, we already talked about. That's the binding site. We talked about that. That's the contraction of the bond. To switch to high affinity. Then there's twist, tilt, rip, and pop. I'll tell you why I call them that later on. The phi gives sequence. It also gives the landscape. It turns out you can calculate the energy landscape from the phi values here. This is calculated from these five values. I call this the transition state ensemble. And this little double dagger, there's your transition state. That's the point that's lurking inside that barrier where you're measuring everything. You're measuring everything by this thing. It's also called the committer. If anybody's into kinetics, that's the committer. You can calculate the committer from this map of phi values. You can't get absolute times, but people have recorded a very short gap of about 5 microseconds. And based on that gap, we can get the ratio of the barriers out of each of these intermediate states. I use a value of 300,000 per second for K to give me this distribution of times across the intermediate states, which mimics that which you would see, which is seen in real experiments. Of course, you can't see in real experiments, about 10 microseconds is your time limit. So you just see this rising phase. You don't see all these components here in experiments. Those are calculated. And the third thing is, oh, there's the committer. And the third thing is phi gives you location. This map gives you location. So we're getting sequence, landscape, and location. And the reason is this. This is a mutant. Each one of these points is a mutant cycle analysis. So we make a mutation of one residue, a mutation of another residue, and then we make the pair. and we say, is this, how much does the energy deviate from the sum between the two? And this, in this orange bar here is sort of our noise level. That's 2 kT. That's what we consider our noise in our experiment. And it turns out things that are separated by more than 20 angstroms, for the most part, do not touch. All the coupling is due with it's close range. So things that are far apart are not more than 15 or 20 angstroms are basically not talking to each other. So they're independent energy changes from mutations of these side changes. There's a few things here that we don't quite understand. So what that means is we can make a map. So this is a map of all the phi values on the protein. This is from the alpha subunit repeated 5 times around. And this is on a different structure, a homologous protein, because at the time we made the map, there weren't any structures of the acetylcholine receptor. And what the white ones are mutations, are plots where the range was too small to get a slope on those, to get a phi value. So we measured all of these things, but we didn't get a phi value because we didn't get enough of a change in energy. So here are the 5, and here it is spread out. The purple ones are the 5.95, the blue ones 0.8, 0.6, and 0.3. And just to make there's a little bit of noise in there. There's these guys are interesting in here, but in general, these are the four domains for those 4 colors. Because I'm going to try to make things simple now. And this just shows you the brown guys, the phi of 0 guys were this is the gate region here. They seem to be clustering at the gate in non-alpha subunits. But I won't go through those now. But so here's our idea, the gating conformational wave. Because we have sequence and we have structure, that this is the sequence of changes in the going from closed to open. And if we add now that reaction coordinate where binding and gating is connected, we can now put catch and hold in there in this sweep. So that's the opening and that's the closing. That's how it works in that order in the protein. So there's this longitudinal conformational wave of opening and a longitudinal conformation range in the reverse direction for closing based on that phi value analysis. I would love someone to compute this. No one has been able to compute this. This is beyond computational level, I think, right? People have not done this. For some reason, I don't know why. So here's it now. I'm putting this together with the zipping model. So here's a closed channel that's going to zip by sequentially changing the barriers along the way until you get an open channel. And this is the other key point, which explains the result about binding. If we start with an open channel, if we start with a channel that's that one. I did it again. So now we're going to start with a channel that's been made to be open by virtue of a mutation. You can make the channel open constitutively, but it's now going to go through the reverse. This is this one. It's going to go through the reverse sequence of events, and catch is going to happen. So catch happens in unliganded receptors. So you switch from that profile of binding to that profile of binding because you unzip across binding and gating. So you zip across catch and hold, and you unzip across hold to catch. So here's the zipper. That's how I think the transition state is changing sequentially. Of course, this happens in a microsecond. This all happens in about a microsecond. That's what the time is. So I'll do that until you get this. I'll do that again. The sound's not working. So that's each one of these: catch, hold, twist, tilt, rip, then pop. Pop is the fusion of the water barriers. So that explains. This model explains those 3 unexplained, all those results. So the catch and hold reaction coordinate and the binding profile. And just as a final to end, I'll just use with this idea in case anybody has an idea. The AChR is a molecular device that effectively transits a few water molecules across a 5 nanometer low dielectric gap. So you have a few water molecules here. This is dry. So you go from wet to dry and dry to wet. is what happens during gating. I don't think it probably doesn't mean anything. It's just something that I noticed, that you're moving about the same number of water, about 10 water molecules. So when you bind a ligand, you displace 10 water molecules. So you lose water. At the same time, you gain about 10 water molecules at the gate. So you're basically moving 10 water molecules across, not the same 10 water molecules, but you're effectively transferring them. So here's the summary, 3 experimental results. Anybody ever says that they figured out how this receptor works, ask them how they explain those 3 results. Because these are solid experimental results. Everybody ignores these, but they are real results. The correlation led to efficiency, classes and modes. The binding gave me the catch and hold idea, which led to the idea of a conformational sweep. There is not binding, there is not gating, there's a sweep. This guy led to the idea of a conformational wave, which had a map, which combined with a sweep gave me a zipper. So that's the summary of that. I'm going to thank people now. I don't want to go too shortchange here. I know I'm late, but Snail, JD, and Prasad Purohit were the first to get us going on efficiency. Although they didn't know it was efficiency, they got the correlation of energies, binding energies. Tapan Nayak did a lot of work on efficiency, and Dinesh and Durfee is now doing the dose response analysis of efficiency. A lot of people were involved in the conformational wave, as you might imagine. If we did 3,000 mutants and all analyzed at the single channel level, Claudio Grossman was at the very start of that, along with Ming Zhou, who was a graduate student in the lab, and Sudha Chakrapani, also a graduate student. And all these other people, I won't go through them all. They basically, I gave everybody a piece of protein and said, what's the phi value over here? With, phi value, and that's how we put that map together. The simulations were done by Sushri Tripathi and Sri Rupa Chakraborty, who's now at Northeastern. Sushri's at Intel Corporation. Wenzhen Zhang was our advisor and all that. And now Pooja Goyal is now taking over that if we can get funding for it. These are the other projects that I worked on in the lab that contributed. And I had many good consultants. John Richard and Rob Colson were the guys that got me thinking about linear free energy relationships. And Auerbach is my son, not the guy that's sitting in the back, my other son, who one day at Thanksgiving, I said, I have this ratio of energies. What does it mean? He's a civil engineer. He says, energy ratio, that's efficiency. So that's where I learned about efficiency. That's where I started to go efficiency based on talking to a guy who builds wastewater treatment plants. And I've had outstanding technical support, excellent funding throughout my career, and I have a bunch of lectures. I'm planning to put more of these out on the web as a sort of a video pamphlet. There's a few out there now. And I finally want to thank the acetylcholine receptor. This guy is unbelievably friendly. This is the most user-friendly protein you can imagine. And all this stuff was first done. And you can add to this Galvani in 1798, who discovered bioelectricity. That was an acetylcholine receptor. So all this stuff first came from the acetylcholine receptor. As I said, I'm not sure about the wave and the zipper. I can't, but I think catch and hold is going to be. I should say that all agonists bind slower than diffusion to the closed state. So it's not just the acetylcholine receptor, although nobody has measured the open state binding. But even if it's just affinity, efficacy, and efficiency coming from the acetylcholine, that's pretty good resume for that receptor. So I have to thank this guy. And I thank you for that. There's the summary again. Well, that's all I want to go. Okay.
Wonderful, wonderful talk. Can you open the shades, guys? And turn on the light, maybe, Ali. Thank you. This was really interesting. I mean, I hope all the students saw how much data goes into these studies and try to do. So I have actually one question. I think that's what everybody was scratching their heads about. You are a structural people. You know, you have a device. You know, the one side binds the ligands, one side opens, and it's a channel, and you can read how many ions go through. So then I'm still actually thinking about this idea of efficiency. What kind of a structural basis? I mean, you mentioned you would like to know from us what ideas we might have, but can you even speculate? I mean, what happens in terms of the ion channel activity of that device that we can define as efficiency? You know, potency is, we know this is the binding affinity. This is how much it takes to occupy. And then efficacy, we know how much response you get from the protein. But efficiency, we have a hard time to fit because maybe we can't define a conformational basis for.
Well, if you allow me to speculate. So I'll
Yeah,
speculate.
please,
I
please.
think that the binding pocket shrinks. And you can imagine springs attached to that binding pocket. And they pull on other parts of the protein.
Right.
Which eventually leads to twist.
Mhm.
So you have hold, the next step is twist. Okay,
Yeah.
in this zipper. It goes
Right.
catch, hold, then twist.
Mhm.
The coupling between hold and twist is an energetic coupling. There's some energy coupling between these two.
Mhm.
And my lower efficiency ligands do not generate that enough, the same, they generate less energy for coupling into the next step of the process. Is what I would say agonist efficiency.
I see. But then at the end of the day, whatever happens here somehow is differently coupled to the ion channel part.
Once
No.
you get to twist, it's all the same.
Oh, I see. I
It's-
see.
You just measure this, we're measuring this. But I think the agonist efficiency is between hold and twist, is what I think, and everything else is the same afterwards.
Right.
But you just, the first step of the gating process is less efficient, you don't get as much energy, and you don't, it doesn't happen with the same probability or the same energy coupling.
Now, I get that. I get that you can have a different conformational response of your protein to different
Yes.
ligands, but you're measuring ion channel down there. So somehow the coupling between all these acrobats in the
Yeah,
ligand
but-
binding domain
Yeah,
and
but
the ion channel is what you're measuring, right?
Yeah, but I could measure the, so, as I said, the GPCR is you're measuring not only the GPCR, G protein, you're measuring that G protein binds to
Sure.
the potassium channel, and then ions get through, all that stuff is the same. I'm measuring what's different for the agonist, and all the agonist cares about is catch and hold.
Okay.
And hold is, and all twist cares about is hold and tilt. You only care about the things that are next to you in this sequence, is the way I
Okay.
think of it.
Okay.
And I think they're all coupled together. I mean, the efficiency is coupled to the
Mhm.
conformational wave. You
Mhm.
have to know all that stuff if you
I
really
see.
want to know that.
I see. I see.
But I will say, if efficiency is just a number, it's like,
Yeah.
there's an efficiency for a gun. When you shoot a gun, that's the same thing. That's chemical mechanical conversion efficiency.
Coupling. Yeah, sure.
35%.
Yeah.
So you can ask, what's the structural basis for that efficiency of a gun or of an automobile? Burning of a piston causing the wheel to turn. That's about 50% efficiency on a good car.
Mhm.
So you can ask, what's the structural basis? There is a structural basis.
Yeah.
But I don't know whether it's the
Right.
piston going up and down or the
Yeah,
transmission.
what
or
part
the
is the-
wheel. There's all
sure.
these different parts.
Yeah.
At the end of the day, you have an efficiency for
Sure.
the machine.
I see. I see. I see.
Does that answer the question?
Yeah, well, yeah, that's, I wanted to know what your thoughts are, actually, on this. So I don't want to dominate the conversation here. Questions? Also from the audience on Zoom, in case you have question, please raise your hand and, or just speak. Claudio, others. Oh, Andres.
There's no audience on this, I don't think.
Oh, so it's following
They've all
this
left.
one, actually. I'm going to put it here.
We did a lot of experiments, but I'm curious, is there a dependency on salt concentration and efficiency? Or just-
Salt concentration and efficiency.
Yeah, because I'm assuming different salt concentrations may change the affinity of the ligand or maybe pocket size. So I'm curious if you notice anything like that.
We haven't measured that. I would tend to doubt it. I think the efficiency is going to boil down to the shape of the pocket. And maybe salts will change whether the pocket goes from this shape to the 42%, or maybe it goes to a different one, something like a 38% when you have a different salt concentration outside. It's possible. We haven't measured that. But yeah, I don't have any information on that. I think, I would like to interest people in computing some of this stuff so that I don't have to speculate and make these wild statements about the pocket changing. And then, I don't really know. As I say, I can show you cartoons of what I think is happening, why loop C. So I think when loop C covers the binding pocket, it pushes the other subunit away. It opens up a pathway for the ligand to get in. That's why things are faster in the open state because loop C does cover the binding site, but it's not a steric barrier. It's actually something that allows the ligand to get in. We have measured the distance between the tip of loop C and a proline on the other binding site, and it gets longer between C and O. It gets further apart. So there are strange things that happen in these simulations that we don't know what the explanation for. And it would be very nice to have someone who was really seriously involved in understanding what was going on. And there's more and more structures coming out every day. When we did this, there weren't any structures. We had to use homology models and things like that. But now there are structures of these things, and they can really be taken on. And we have this magnificent set of data between this C and O. If you can figure that out. And here's another point for computational people: the off rate is the same in closed. So a lot of people measure off rate for ligands and think they're measuring affinity. Affinity is in the on rate, not in the off rate. So it's a mistake. People scan billions of compounds. I heard a seminar about scanning 1 billion compounds by measuring the off rate. And I'm saying, dude, it's the on rate that determines the affinity. So this is important stuff that I think, and I think this is also true for GABA receptors and glutamate receptors. So I think this is a common thing. So I'm trying to get people interested in doing computation on this project. So there's an excellent data set which you can use to compare your numbers to.
Nice.
So I know I'm talking too much.
Sure.
Any other questions?
Zoom people, do you have any questions?
Yes, hello, I have a question,
Go ahead.
on the Zoom. Do you hear? Thank you for nice talk. I'm really interested to know that what is the role of the transmembrane domain on ligand binding in the catch and hold, because I'm asking about this because I want to know, can we just study the extracellular domain of the nicotinic receptor for binding affinity, something like this?
All I can tell you is that we've made hundreds, if not thousands, of mutations in the transmembrane domain, and so far we haven't found anything that changes the coupling constant. So there's very small effect on the the elements in the transmembrane domain on the coupling constant. We don't know whether they change the closed affinity and the open affinity to the same extent, but the ratio of those two affinities stays the same. So I think that there's going to be no effect on the affinity from the transmembrane domain. We have yet to find a mutation that changes the affinity in the transmembrane domain. There are reports in the literature, and these reports are incorrect. There are mistakes in the Bible. People have reported them, and we've reproduced them, and they're wrong. So it's tricky. It's hard to measure these things correctly, and if you measure them correctly, we find there's no effect of the transmembrane domain protein anywhere in the protein. There's 4 helices per subunit, and we've mutated, I would say, almost every residue and almost every helix, and we have yet to find one that changes affinity.
It's very interesting because I simulated the nicotinic receptor, just the extracellular domain, and for the resting state or closed state, low affinity state, I could detect that the loop C wouldn't is not stable and tend to close the binding site, and the resting state quickly turn and change to the desensitized state.
This is computational or experimental?
Computational.
Well, okay, it's wrong.
Oh.
I mean, it doesn't happen in cells in real life. You don't see a change in affinity when you make a mutation in the transmembrane domain. Now, I don't know. I think the resting state is very unstable. In our computations, it immediately switches to desensitized or open state. Desensitized and open state have the same affinity. So when we do computations with a closed channel, it very quickly, within 20 nanoseconds, it flips to being desensitized. And so I'm not surprised that the C state is very unstable in computations. And I don't know why that is. But in experiments, I'm just saying, if you take mutations and you measure KD, specifically measure the coupling constant, you see no, I should be over here. You see no, you don't see that experimentally. So I don't know. I don't know why you switch to desensitize quickly, or when you make mutations of transmembrane domain, but it doesn't surprise me because the resting state is quite unstable in our computations. It very quickly switches to high affinity. The binding site switches to high affinity very quickly in our hands too. So,
Where are you, Mina?
In Iran. In
Oh,
Tehran.
you're coming.
oh, okay.
Yes.
Okay.
You
Wow.
know, and I have another question: that do you have water molecules in the binding pocket for the resting state?
Yes, there's a
Yes?
few. Yeah.
Oh.
It's not completely dehydrated. I can't remember. I don't remember. As I said, the agonist displaces about 10 water molecules, or I don't know how many, but I think there may be, it depends on how you define the pocket exactly, but there is water in the resting binding pocket, but not much. I don't remember the numbers for that. We did that in one of our papers.
Okay. Any other questions? If not, please join me to thank Tony one more time.
Thank you.
Fascinating, really.
It's a ton. It's a lot of data.